So just imagine you're at home, you're in your living room, and you have 5 virologists on the couch. Well, to have a conversation, that is. And that's what this is all about. This week in virology, episode number 102, recorded on October 7th, 2010. Hi, everybody. I'm Vincent Racaniello, and we're in Munich, Germany for a conference called Viral Offense and Immune Defense. Oktoberfest may be over, but we still have Weissbier and Helles. Is that right?
Yeah, it's right.
So this is a special TWiV coming from Germany today, and I have 4 terrific guests to join me. On my right, Katharina Eisenacher, is that right?
Yeah, it's right.
Who's a postdoc in the lab of Dr. Anne Krug at the Technical University of Munich. Welcome to the show.
Thank you.
And we call it a show, even though it's science, because that's what podcasts are called. We'll have a few more words on that later. Next to her right is Karl Klaus Konzelmann, professor at the Ludwig Maximilian University right here in Munich. Welcome.
Hi, nice to be here.
Thanks. To his right is Andrew Baker, who's a professor of molecular medicine at the University of Glasgow. Welcome to the show.
Thank you very much.
And finally, my scientific father, all the way to the right, Peter Palese, professor and chair of microbiology at Mount Sinai Medical Center in New York. Welcome.
Thank you very much.
It's a revisit for you. You were on one of the shows way back in 2009. We did a special show just after the influenza pandemic emerged last year, and Peter explained it all, and we had a lot of interest in that show. And also welcome the audience. There are about 10,000 people here, I would say. So, by the way, this is a conversation, so everyone can participate. If you don't understand something that Katharina says, you can ask her, and vice versa, and you guys too. And I'll tell you what, to motivate you, I'll actually come up with the microphone. Anyone who asks a question gets one of these really nice magnets. It says, microbes, we need them. Okay? You can put it on your refrigerator or whatever. I have 4 of these. So if you ask a question, and no question is stupid. So what is this that we're doing? This is obviously unusual for a scientific meeting, and I'm really happy that I was asked to come here and do this. Now, TWiV, or This Week in Virology, is a weekly conversation about viruses. I usually do it with 3 or 4 co-hosts who are always on the show. And I'm sorry they couldn't be here today. They would be Dixon Despommier, who's a professor at Columbia, professor of parasitology. Alan Dove, who is a former student of mine, a PhD student who's a science writer. And Rich Condit, a professor of microbiology at the University of Florida, Gainesville, and an expert on pox viruses. So every week we select stories about viruses and we talk about them. We do this via Skype, and I record the conversation. It's released every week on the internet around Sunday, and it's downloaded. And we started with 0, and now 2 years later we have sometimes 15,000 people downloading each episode. And we have a lot of interaction with our listeners. And at the end of the show, typically we read some of the emails, and we'll do that today. And you can see what fantastic fans we have. We have people from all walks of life listening. There are many virologists. Anybody here listen? Can you raise your hand? About 5,000 of the 10,000. Very good. And we have physicians, we have physicists, chemists, we have people who don't work, and as you'll see, we even have truck drivers listening to TWiV. So the power of this is that you can make it very easily yourself. You can do this. It's a little complicated here because we're doing it in an auditorium. But you can do it. You can release it. It's free, and people can get it for free. So it's a very powerful way to spread information. And I decided to do this because I'm passionate about viruses, and I wanted to spread as much information as I could. So today we'll have a conversation with these wonderful folk here who have decided to join me, and everyone will listen to that. So the information we produce today can be listened by anyone all over the world. So that's the power of a podcast. So I'm really grateful to Jens in particular, who made this work. We were missing a cable at the last minute, and he found one, an adapter, so thanks very much. I want to tell a story about Jens. He picked me up at the airport the other day. So I was looking forward to coming here because I have a long commute, and I said, boy, I'm not going to have to drive and sit in traffic and for a whole week here in Germany. So he picked me up at the airport, and what happened? We got stuck in traffic. So he wanted to make me feel at home, I guess. And then we were late, so we had to take the subway. So again, he wanted to make me feel like I was in New York taking the subway. Okay, let's get on with the real stuff here, since you're on my right. Are you nervous?
A bit.
You shouldn't be. You're very brave. So you're a postdoc, right?
Yeah, but it's a kind of strange situation.
Strange because you're in front of a audience? Yeah, but when you give talks, you're in front of an audience.
Yeah, but
also.
then I stick to my science.
Okay. Well, you should get used to this because I think this is the future of teaching. But you're going to be fine.
Okay.
Because you know more than we do.
Hopefully.
For example. So you, first I wanted you to explain, I called this the SFB, right? SFB 455,
Yeah.
which is, and it's the junior faculty of that which is running this meeting. Why don't you tell me what that means?
So the SFB 455 is a collaborative research center, which stands for SFB, and they all have numbers. So this is a very popular scientific, collection project in Germany. So usually, so you apply for funding, and then different groups and different institutions come together and they form such a center. So it's kind of interdisciplinary, and the 455 is just our number, and our topic was viral function and immunomodulation. So in our SFB we have virologists, we have immunologists, we have structural people, so and we are interacting together to build a really excellent science unit, and we exchange methods, viruses, plasmids, and we do retreats together, we share thoughts and discussions, and I have the feeling it's more easy to do good science in such a environment.
So in German, what is SFB?
It's Sonderforschungsbereich.
Very good, I like that. Our first German on the show. So this is like a countrywide, almost a program project grant, if you would, in the US, right? That would be the equivalent. And it's ending now, I understand.
Yeah, so we had funding for 12 years. We were lucky because maximum funding period is 12 years, and you after 3 years you have to show your data, and then it's possible that you get funding for 3 more years, and we were really happy because we made it until 12 years. But unfortunately now it's really ending, and that's why we are here.
So the meeting is sort of a celebration of the 12
it's the final
years,
party.
And Klaus, you were part of this 12 years also, the whole 12 years?
The whole 12 years, I'm one of the few who started up 12 years ago as a virologist, and that, as you have heard now, I'm now a neurobiologist and immunologist.
I heard, yeah.
That's the advantage of such an SFB, who brings together, as she said, different disciplines, and it provides the possibility to do some projects which you, with your own group, which is funded for 2 or 3 years, you would never accomplish.
I don't think we have anything quite like it in the US. A program project grant is typically within an institution. What about in the UK? Do you have?
No, nothing that you can carry on for that long, 5 years at the most.
Yeah, and also
Very rare to get anything longer than that.
I mean, these groups spread across the country, basically, right? In theory, they could.
So it's more local.
This is local?
Yeah, so it's the Munich area, and there are other SFBs, but they are in different areas. Then there are also other SFBs in Munich as well, but also spread over Germany, but then locally.
They're locals, but they are multi-institutional.
Yeah.
So that's different. Most such programs in the U.S. aren't like that. So that's the SFB, and they've supported this wonderful meeting, and these are some of the people who have been speaking at this meeting. So how did you get into virology?
I started biology, And I studied biology, but to be honest, I never was that much interested in this zoology and botanics and all that stuff. And that's why I specialized into immunology. And then I did a diploma thesis in virology, and this was the first contact with the virus. It was rubella. And then I thought it would be cool to combine immunology and virology. And that's why I was looking for a thesis, a PhD thesis, where I can do such a project.
Right. So what have you been working on in your postdoc?
Now I'm working on RNA helicases. I do some RIG-I stuff and some innate immune regulation stuff, and therefore I need the viruses.
Right. So RIG-I think we've talked about it on the podcast before, is one of these cytoplasmic proteins that senses RNA. So exactly what have you been doing with RIG-I?
So within this SFB, we identified a structure at the C-terminus of RIG-I, and this was quite novel because until that time, it was thought that RIG-I consists of an N-terminal CART domain and the central helicase domain, and we identified this regulatory domain, and within this domain you have a triphosphate binding site which specifically detects the pump, so the pattern that is the danger signal in the cell. And this revealed some new facts on the RIG-I pathway, how it's activated and how downstream signaling is recruited. I
think I remember reading in your poster or abstract that you had found a new dominant negative form of RIG-I, is that right? Yeah, I have some hints that there's something new, but until now it's really preliminary. So it looks like some endogenous regulation. There is already a related protein that I think it's LGP2 it's called? Yeah. Which is missing the CART domain? Yeah. So it will bind RNA, but it doesn't signal, right? Right. And that's thought might be a negative regulator. Is this a similar
So the
protein?
LGP2 is a negative regulator, but it's also an RNA helicase, so it belongs to the RIG-I family. And what I found is that RIG-I itself, by inducing a kind of short form of its own protein, negatively regulates its own pathway. So I have the feeling that it's a kind of control mechanism to balance the innate immune response to prevent this overwhelming interferon induction.
So is this produced during splicing or proteolysis?
Yeah, this is the issue I try to address now. So I'm not really sure. I know that it's there, but I don't know the mechanism.
Okay. How much more time do you have in your postdoc?
I think there's some time. I hope that my PI is prolonging my contract.
She's smiling. Some time, a couple of years?
No, not a couple of years. Hopefully, if I find a mechanism, then maybe it will be finished next year.
And what do you do next?
So I like to be at the university and in the lab because it's so you can wear casual clothes. You can come there in chucks. So I think I will do another postdoc and not this serious industrial stuff.
Yeah, we certainly are casual in labs, that's for sure. But it's also attractive because you can think about what you want. Yeah. Which is better than you can do in many other professions. Anything else you want to tell us? Where are you from, by the way?
I'm not from Munich originally, so I was born in the eastern part of Germany before the wall fell down. I don't know if you know that by chance. The city is called Leipzig.
Sure.
It's close to Dresden, and I studied there, and then I moved to Munich.
So it's fair to say that if the wall hadn't come down, you wouldn't be here today.
That's true.
So it's a good thing because science flourishes when people work together. Yeah. So that's a great story. All right, well, thanks. And do participate if you hear something that you don't understand.
I try.
For the rest of the talk. I want to move to Andrew now, who I've never met before. This is the first time. This is
nice to meet you.
Interesting way to meet someone at a podcast like this. But I read in the summary of your work online that you work on using adenoviruses to do gene therapy for cardiovascular diseases. And we're going to hear you later, but the audience who's listening now would love to hear what kind of cardiovascular diseases are you working on?
The gene therapy side is focused on coronary artery bypass grafting. So when patients come in with angina, they either go for a coronary intervention, which is a stent, where it's a metal scaffold that will hold the coronary arteries open, or they go for bypass grafting using, and because there's usually multi-vessel disease, patients need multiple bypasses.
Mm-hmm.
So if a patient has one bypass, you generally use the radial artery because it's an artery to an artery bypass graft.
That's from your arm?
Yeah, or internal mammary is the other option. But because most patients require multiple bypasses, that's not enough material. So saphenous vein from the leg is used.
Mm-hmm.
And the problem with that is you're putting a vein into an arterial circulation, so it has to try and adapt itself. It tries to turn itself into an artery very quickly to cope with the higher blood pressure. So that works okay, and you get the media of the vessel wall thickens so it can cope with the higher blood pressure.
Mm-hmm.
So that's adaptive remodeling. We also have this process called pathological remodeling, which leads to reocclusion of the vein graft. So the patient over time will come back in with angina. And considering there are 500,000 operations worldwide and 50% of them lead to a reoccurrence of symptoms, there's a big impetus in trying to find some way of preventing that. The pharmaceutical industry has tried but has not come up with anything useful. So we thought, because you're taking the vein from the leg and putting it into the coronary circulation, there's a little clinical window where you could genetically modify the graft to try and prevent that pathological remodeling.
Mm.
So we use the adenovirus during that 30 minutes to infect the vein and express a therapeutic gene, which is a metalloproteinase inhibitor.
So the vein is taken out and placed in a dish for half an
Yeah.
hour, and then
Yeah.
you infuse it with adenovirus.
You let it sit there for half an hour,
Oh.
and then you wash it free of any excess, and then you graft it.
So the metalloproteinase inhibitor prevents the pathological remodeling.
Yeah, it does. In the models we've tested it in, there's a human ex vivo model where you then just subsequently culture the vein,
Mm-hmm.
or there's a large animal pig model that we've used, and it works very effectively over a long period of time. Although the virus doesn't hang around for longer than a month, you get a lasting benefit because the pathological remodeling really happens during that first month. Okay, so it's quite good the virus actually subsequently goes away.
So that's a case where the acute
Yeah,
adenovirus infection is perfectly suited
yeah,
for that.
yeah.
So have you tested these in people?
We are waiting. On the 1st of November, we have a funding decision from the
Mhm.
Medical Research Council in the UK to do a phase 1/2 clinical trial. So we'll hear about that.
So you have GMP produced virus ready to go.
Very expensive, but
Yeah.
it's there.
Yeah, so in the US it's $5,000 a day to rent a lab to do GMP.
Yeah. So this trial will be total cost of 4 million,
Wow.
and the virus production is 1.1.
How many patients are involved?
160. So in the phase 1, we'll do dose escalation in 4 patients in each group, and then once we know that that's safe and well tolerated, and then there will be 60 to 80 patients in 2 arms. We probably will have an internal placebo control, so
Mhm.
we'll have patients who are grafted twice, one without virus, one with virus, but both with the vein from the leg. So within each patient you have a placebo control.
So in the half hour that you do the infection, the infection initiates. Then as the vein is grafted, the infection's continuing for at least a few days or a week or so, right?
Yeah, the virus will hang around. You can still see a little bit there at 28 days, but the vast majority is gone. It's there for using the first generation adenovirus. It's there maximally, but during the first week, 14 days, and then disappears.
Which serotype do you use?
We just use Ad5.
5.
Yeah.
So is there-
although in future we may switch from that.
Is there any problem with people having antibodies to Ad5?
In the clinical trials that have been done in other cardiovascular and other diseases, then there doesn't seem to be a huge issue. I mean, if there was, we'd have to exclude the majority of patients.
Yeah.
And then would have to switch serotypes or develop a virus that didn't have wasn't neutralized,
Right.
which we could probably try and do, but we don't think at this point it's going to be a real issue. But we will look at that.
Now that the three-dimensional structure has been solved at high resolution, we now can, we just talked about this in our last episode. The neutralization antigenic sites are very clearly resolved on that structure where they hadn't been before. And one of the comments in the paper by the authors was that maybe we can modify these viruses antigenically. So you can, in theory, make one that no one has antibodies against.
Yeah, sure. I think we'll do that. We've also would want to add into that modifications to the virus to make it more infectious, because that sort of 30-minute clinical window isn't, there aren't many viruses that are efficient enough to deliver to the cells during that 30-minute clinical window, because most people, when you do anything in the lab, you leave it on overnight and then come and wash the media.
Yeah.
But during this 30 minutes, it's quite tight. So Ad5 is quite efficient, but you still have to use quite high titers. But we have ways of modifying it that we can improve that uptake quite effectively. So any sort of second-line virus that we take through would have, hopefully, reduced neutralization, but also increased infectivity capacity.
Are these vectors that are can only replicate it, get in the cell, express, and not produce any more virions,
so they're
right?
just E1 defective. Yeah.
Right. So there's no problem with these viruses getting into the circulation, which they would do, right?
Not in this particular condition, because before you graft it, then we can wash it free of any excess virus. You may have a small amount of virus that's bound, that's not washed away, and then it's when it's grafted, it's released, but would be very small. In other cardiovascular trials, there's a lot of catheter-mediated delivery, so you advance a catheter to the coronaries and then release virus through the catheter. In that situation, you do get a lot of release into the circulation, and the virus will probably end up in the liver and the spleen.
But we don't think there'll be any problems with having a metalloproteinase inhibitor in other tissues, though, right?
No, I don't think so. We have tested that, and we don't see any real toxicity from that.
So in the patients in the trial, what will you look for? Long-term for angina would be the readout of it failing, right?
Well, you can't do that because it's really quite chronic, and you'd have to have the trial running for about 5 to 10 years.
I see.
So we'll do quite detailed imaging at 24 months, at 12 and 24 months, using imaging techniques, and that will measure the neointima. So the issue is that the patients who come back in with angina have a lot of accelerated atherosclerosis in the graft, and that's initiated by this smooth muscle cells that migrate from the media, forming an initial lesion. So it's the initial lesion that we'll try and measure at 12 and 24 months, and that we'll try and prevent using the gene therapy.
How do you measure that?
You can just use imaging techniques that give you the 3 dimensions of the structure of the vessel, and then you just quantify that. It's quite simple.
That's great. It's non-invasive, right?
No, it is invasive. Yeah. So you have to use a catheter.
Put a catheter inside.
Yeah.
So the trial will take 2 years, essentially?
Yeah.
And at the end of
Well,
that,
it'll take a while to recruit, and then it's 2 years follow-up.
right. And then if it looked good, say if there were no pathological remodeling or less, then what would be the next step?
The next step would be to secure someone to take it forward into a phase 3 trial. I mean, that would be the big thing, and then for any cardiovascular intervention, it'll have to be tested against, a lot of patients in a phase 3 study. So that would require a lot of other effort that we don't have, and interactions with industry that we don't have at the moment. This is all Research Council funded and charity funded so far.
Is there a big potential market for
Huge.
this?
Well, there are 500,000 operations each year.
In
the
the
beauty
UK
of it is we're not changing clinical practice
All right.
at all, okay? Because every time something comes out of the leg and it gets grafted, so you're just changing that little bit of it.
There are 500,000 in the UK,
No, 500,000 worldwide.
globally.
Yeah, there's about 40,000 in the UK.
Can I ask a question?
Of course.
So when would you call this a success? So what is the criterion? How much reduction in terms of the arteries being clogged are you expecting?
Well, from the preclinical data, we'd expect at least a 50% reduction. So, at 1 year. So the difficulty is then knowing how that impacts on the subsequent atherosclerosis and those patients coming back into clinic. So the ultimate thing is, do less patients come back in with angina? Because doing re-interventions in people who've already had coronary bypass grafts is actually very difficult. So that's the economic benefit, is stopping patients coming back in. But quantifying that will take a long period of time. So we imagine we'll get a reduction in neointima of 50%, we'll be happy. But we'll be very happy to make sure it's safe in the first place. And then we'll go forward for the efficacy.
I would be interested if the people, so the patients you are treating, really trust you that they don't get infected?
I mean, that's, you know, in the UK, then the Gene Therapy Advisory Committee and the ethical regulation around all those trials, patients are very well informed, they're consented, they have patient
Yeah, because usually you have the feeling when you tell somebody, I'm going to treat you with a virus, then they
yeah, you have to get over that
skeptic.
initial, it's for the first few patients until the next patients know that people have already been injected. I don't see that's a problem. Recruitment into other trials has been okay. There's been a problem with pro-angiogenesis trials in the cardiovascular field, just because of that angiogenesis effect that you may have pre-existing tumours, and you know, recruitment's been quite difficult into those because of the exclusion criteria are so broad. But I don't see a major problem. In the west of Scotland, we have a very sick population. If you look across Europe, I think we have the worst cardiovascular risk and worst cardiovascular events per population in the whole of Europe. So we will be able to recruit 160 patients in, well, if it was every patient that consented, it would be in 80 days. But I can't imagine that's going to happen. So we've got a lot of patients.
Okay, and is there any possibility to kind of measure the expression of the metalloproteinase? Because when I think on in vitro or doing some mouse experiment, you would go for a tagged one and then you measure GFP or whatever.
Yeah,
but in the patient it's difficult, I guess.
It is difficult because obviously you can't take out the tissue that you want to prevent the graft failing in. But what we will do is when we do the procedure in the dish, not all of that will be used for the graft, so you can take a little bit back to the lab and make sure there's virus in it.
Because
a few
after
days
30 minutes
later,
it's too early to measure anything.
Yes, you will culture it for 4 to 7 days and then make sure that there's virus in the graft. So that would tell you you have gene delivery to the graft.
So the alternative would be to use animal grafts, right?
Yeah.
From pigs, say, which we should probably comment on, since there are so many endogenous retroviruses in pigs, that may not be such a good idea in the long term, because we don't know what the fate of those would be. So this is clearly better.
Yeah, I mean, there are really no other alternatives, and there are lipid lowering drugs that have a small effect on the level of atherosclerosis that you can get in the veins, but it doesn't impact on the rate of failure. Apart from that, there really is nothing available.
Sounds like a really good application of adenovirus therapy.
Yeah,
And I have to admit that this is the first time I'd heard of this coming to this meeting, so thank you for inviting us to this meeting so we could hear about it. Hope it
Pleasure.
turns out good. Well, we'll be
Thank
following you.
Well, we need to get the funding first. So.
Yes, it's a minor
That's
problem.
the most important bit.
Well, I'm sure you're creative at that. Peter, a number of months ago, you published a paper, and I think you discussed the results today, where you express a headless HA and try to get a broadly neutralizing influenza response. So I wanted to ask you to explain why you chose the stem or the headless HA to do these experiments.
So we are trying to make a better influenza virus vaccine. So this is a situation with influenza where we do have actually very good vaccines, and they are unfortunately not being taken enough in the general population. And one of the reasons is that people have to take that vaccine every year, and this is a problem in terms of manufacturing it. The formulation for influenza virus vaccines has to be changed because the influenza viruses change from year to year, and there are at least 3 different strains circulating in the human population which necessitate to have a vaccine formulation with these 3 different strains. Unfortunately, the decision which vaccine formulation is going to be used for the next season has to be made in February for the following season, meaning there is at least a 9 to 10 month the difference in time in terms of when the date is when the vaccine formulation has to be decided on to when finally the vaccine is being given to patients. So it is, even though we have vaccines, in contrast, for example, to HIV, where we absolutely do not have a vaccine, and it's not likely that it will come in the next year or two. So for influenza, we have vaccines available, but there are complexities in terms of vaccine manufacturing and also the difficulty, logistical difficulty of giving the vaccine every year. So there is in the field a dream to make vaccines which are longer lasting and which may be working against different strains which are circulating. And we refer to this as a universal vaccine, which would be good in terms of protecting against the 3 different strains which are circulating, and also hopefully in the years to come. So what is sort of the thought we have in terms of making such a universal vaccine? And as you mentioned, we try to modify the hemagglutinin, which is the major antigenic protein on the surface of the virus, by changing its sort of appearance or its structure. What we recognize mostly, or our immune system recognizes, is the tip of the hemagglutinin, and that one varies quite dramatically from year to year, and that's the reason why we, when we vaccinate with one strain, and 3 years later, if the virus comes along which has changes in the tip of the hemagglutinin, then our immune system doesn't recognize this new virus or recognizes it to a lesser degree. And so we thought maybe we can make a vaccine which doesn't have this antigenically important site at the tip, but rather only provides the stem, the shaft, the part of the hemagglutinin molecule which is actually conserved. And so what we tried to do is we removed the head, and there happens to be a disulfide bridge which one can bridge directly with glycines and remove actually this whole tip which consists of this immunodominant portion. And so the hope is, and we have only mouse data at this particular point, the hope is that one will induce, by giving as a vaccine this shaft or this headless construct, that we as people would make an immune response which would be effective against these conserved regions. And since these conserved regions don't change from strain to strain, the hope would be that we could make a vaccine which would induce an immune response in the patient for a longer period of time, 5 years, 10 years, maybe even longer, and that we would have a vaccine which is similar to a measles vaccine, similar to a mumps vaccine, similar to rubella vaccine, which lasts, if not the whole life, but at least half the lifetime.
So in that paper, you showed cross protection against which subtypes?
So we tried to have an H1, and we also were successful in terms of H5. So we were showing that there was protection in mice again.
Right.
That we can only protect mice, and let me just hasten to say, it is always easier to protect mice against probably a lot of things, but certainly influenza, than it is humans. So in this particular, in the challenge model which we are using, we were able to protect against 2 different subtypes.
So what was the immunogen from H1?
It was H1, yes.
And it protects against H1
and H5,
H5s.
yes.
So did you try ferrets? Because I understand that's another.
So we, so in general, particularly for the FDA in the US, there's the 2-animal rule. So before one would go into humans in a trial such as this one would have to show that it doesn't kill two different animal species, so it's safe, and two, that it is effective in two different animal species. So mouse would be one, and a ferret is the other accepted animal model for influenza, and therefore it would be necessary to do this in two different animals. At this point, we didn't have enough of the material to go into a bigger animal, but that's obviously what has to be done.
So if I may question, the approach you took was to immunize successively against diverse HA types in mice. So what you somehow reproduced was, as a human, you are infected from time to time, every couple of years, with different types of viruses. So does that have the same effect? Are elderly people more immune against influenza virus in general?
So you are addressing, so we were able to make specific antibodies which recognize this stem portion, this conserved one, and that allowed us then to make it an antigen. So we obtained these specific antibodies which recognize this conserved domain by immunizing with hemagglutinins, which are very diverse. In humans, people are always vaccinated, are infected year 1, year 3, year 5, etc. So in that case, they make each time an antibody against a related immunodominant epitope. So this is really very different what happens in nature. If one would only be infected every 20 years, probably it might be that people would also make only these specific antibodies against these conserved sites. So yes, you are absolutely right, but unfortunately we get infected many more times, and therefore we make usually antibodies against only the variable site, which is the tip of the hemagglutinins.
So you mentioned today that you used virus-like particles to deliver these headless HAs. Would that, I think you said it was of convenience to a certain extent. Would that be what you would eventually go into people with, or something else?
So we believe that the protein alone is not immunogenic enough. So this headless construct, even though it is probably a trimer, it is not immunogenic enough per se. And so the first try was that we would put that onto a virus-like particle, which is sort of looks like a virus, but isn't a virus, and has just these headless constructs on it. So we are trying to avoid that we give a regular hemagglutinin. So we could put it on a virus, the regular hemagglutinin and the headless, but that would be probably not effective, because all we would see in terms of our immune system would be the regular hemagglutinin, because it's immunodominant. So we want to provide something which looks like a virus but only has this headless construct on it without anything else.
So you have to solve the yield problem before you can go very far with
We
that,
have
right?
to solve the yield problem, and so this is a collaboration with Adolfo Garcia Sostre, also at Mount Sinai, and we are really thinking very hard how can we improve the delivery, not only the delivery, that's another question, where you deliver it and how, but just making more of an immunogen which is really inducing a lot of an immune response.
So if everything went well, it's conceivable that this form of the headless HA might go into people at some point in clinical trials, right?
We would certainly hope that we would get support to put that into people, yes.
Now, I'm sure you know this. Gary Nabel's group published a study not too long ago where he took the approach which has been tried, I guess, for HIV, is to first prime with a DNA, a plasmid against the HA, and then boost with a recombinant adenovirus encoding HA. And that regimen seemed to provide universal or broadly reacting antibodies as well. What do you think about that?
Yes, I think it's, in a way, it's sort of similar to come with different immunogens. In his case, actually, he used a DNA vaccination and then came with an inactivated virus. And the inactivated virus probably, the tip again, the immunodominant was probably more inactivated than the structure which was made from the DNA. So he was probably boosting with a shaft, again, very similar to our situation, by using two different forms of the immunogen. And by that, he was able to just enhance the conserved structure. So this is sort of another way of doing basically the same thing, namely inducing an immune response against the conserved region of the molecule.
Could be that the DNA part also played a role. You get a different B cell
It could
right?
also be that it has to do with where it is being induced, etc. But in essence, it's the same strategy to boost with something which enhances the immune response against conserved regions.
Of course.
I have a question. So if when you are producing this virus-like particles, is this the same? So is it mimicking the influenza particle, or do you have more of HA on the surface?
No,
so it's a kind of quantitative thing.
Yeah. So I think there is some Zinkernagel once sort of postulated that by having more of the same antigen he called it periodicity, that this would actually enhance the immune response. And so all we want to do really is provide that construct on a particle where we may have a periodicity. I think these gag-like, or these are gag-based, HIV gag-based virus-like particles. They probably don't have as many spikes, as many hemagglutinins as a regular influenza virus. So that's probably not as good, and maybe one has to find a better way of doing this. So there are other ways of thinking of enhancing the immunogenicity of that construct per se. Freund's adjuvant is, for example, one way of doing it, but clearly that is highly immunogenic and is frequently used in animals, but clearly we could not use anything of that nature in humans.
And there are other adjuvants that are being looked into, I think, because they can shape the response and perhaps bias it towards more cross-reactive epitopes. But the public is very wary of adjuvants, as you know from the experience in the past year with the swine flu, the swine origin vaccine. So that may not be the wisest way to go.
So in the US, there is a great hesitation to use adjuvants in vaccines. Whereas in Europe, there is a little bit less. Some influenza virus vaccines have been licensed which have adjuvants. So there are different cultural decisions in terms of regulatory issues.
One of the another podcast not related to ours called Adjuvants Hamburger Helper. Now, I don't know if you have that in Germany, but in the U.S. you can buy this stuff in the supermarket, and it makes your hamburger go farther. You know, if you have a lot of kids to feed and you don't have that much hamburger, you put this in and it stretches it. So he said an adjuvant is nothing more than hamburger helper. But that's wrong, of course, because it makes a different kind of antibody response. But the public's perception is very different. That's why we do this in part, is to try and get the word out, and the more people who listen, the better. I'm actually amazed that we've come so far in such a short time, because we've been talking about universal vaccines for influenza for a long time, and now suddenly in just the past year, has there been one discovery that has sparked all of this, or is it just accumulation of?
I think there is some interest in the field, both in the HIV field as well as influenza, to think about universal vaccines on one hand. And the wish was there for some time, but what has really happened is that both in the HIV field as well as in the influenza virus field, several groups have discovered monoclonal antibodies which are cross-reactive and which are recognizing conserved domains on the antigenically important protein. In the case of the HIV, is the GP120, GP160, and in the case of influenza, it's a hemagglutinin. So the finding that there are really, and that people make antibodies which are cross-reactive from one strain to the other.
Yeah, it's interesting that similar things are happening with HIV antibodies now. I guess it's technology driven, the ability to take B cells and make libraries from them and identify the sequences helps to do that.
So in a way, if we make cross-reactive antibodies, but it's only 1 out of 1,000, it doesn't help us. But if one has a good technology to fish out that one monoclonal antibody, and then we make a vaccine based on that one monoclonal antibody which works, that would be sort of the way people are thinking now, both in terms of influenza as well as in terms of HIV.
So what's the ballpark, you think? How many years will it take?
I never say never, but I think it's a difficult path. I think I hope and I'm optimistic that for influenza, which is an easier problem, that we will be able to get there earlier. But I think it is still a reasonable possibility that we will succeed in making, we meaning the field, universal vaccines against both influenza as well as HIV.
Okay, so you don't want to provide a year. Hey, Adolfo, what do you think? 10 years? These guys are conservative.
I'd be very happy from 10 years.
Yeah? I would say 10 years, but I don't know. You guys are in the field. I'm just curious. It seems to me we're using a very old vaccine, even though it's reasonably effective, and it's time to do something different. But and something that doesn't involve eggs would also be useful. Well, I had one more question for you. Do you want a vaccine that'll hit, say, all H1s or H1, H3, H5, everything? What would be the best?
I mean, we would be very happy already if we have a vaccine, one vaccine construct which would which would protect against all H1s, and the second one which would protect against all H3s. But theoretically, it is possible that we would have one construct which would be really cross-protective against H1s and H3s, and possibly an H16, which may come one day.
Right. That would be the ideal situation.
That would
right?
be the ideal one.
And then something you wouldn't have to take maybe every 5 or 10 years would be, and that would improve public acceptance of it as well.
Absolutely, and make it cheaper, and would hopefully, I mean, theoretically, we could think that, for example, influenza B virus, on which Dr. Raccaniello has worked in the past, we could theoretically eradicate influenza B viruses. Because if we would immunize against influenza B, then the virus does not have an animal reservoir and could not survive without the human host. So it would be very similar to poliovirus, which may drive out some of our colleagues in terms of support, but theoretically we could even think of making a world without influenza B, and a good influenza A vaccine may literally prevent influenza infections worldwide.
Yeah, you have to be careful. If you eradicate the viruses, then you're out of work.
I think that probably would be worth it, though.
Of course, of course. Well, we can find other viruses. There are enough to work on. How are we doing on the film there? 10 minutes? All right. So we're also videotaping this as a special treat. And our tape only lasts an hour, so we have to be conscious of that. So let's move to Klaus Karl and negative strand viruses, which I understand from you are becoming positive strand viruses now. So one thing I wanted to ask you is you were the first to make a infectious clone of rabies virus, the first negative mononegavirales. Can you remember those days and tell us something about them?
Oh yeah, these were of course very exciting. and actually I started to work in virology as a postdoc, and I went to a laboratory where they worked on positive strand RNA viruses. And they said, oh, this guy Raccaniello can make infectious cDNAs, and they did it then for some positive strand RNA viruses, BVDV, or cholera virus, and I was always very jealous what these people could do. They modify, take parts out, put parts in, and we could not do that at these times. But there was great work from Peter's group and also from the measles virus group in Zurich. All negative strand virologists tried to find out how that works, and finally it worked with rabies virus, and 10 years later, we could then also start to modify the virus, which is very important in terms of finding out how this virus works in molecular terms, how it antagonizes interferon, how these viruses could be used to make vaccines to express foreign antigens, and of course as gene therapy vectors, which we will hear more this evening.
That's right. So the unusual aspect of rabies and the mononegavirales in general was that you have to put in a plus strand RNA, even though the genome is minus strand. You have to put a plus strand in, correct?
Yeah, that's very logical, because if the infectious unit, the transcriptionally active unit of such a negative strand virus is a ribonucleoprotein in which the RNA is encapsulated into nucleoprotein, and you have to simultaneously express the viral RNA and then the virus protein to somehow form an RMP-like structure. And if you would express the minus strand RNA as a viral genome, and in addition express the plus strand messenger RNA to make the proteins, that would lead to hybridization. And nowadays, of course, people are aware what hybridization does. At these times, people used already antisense RNA to block translation from a protein, so it was not good to have hybrid. So we just tried with positive strand RNA, and then everything worked. That requires one step more of replication of these novel RNP-like structures, but that apparently made the difference.
So you had tried negative strands for a while and it didn't work, right?
Everybody in the field tried negative strand RNAs, also with them. They tried with mini genome RNAs, and if I remember right, only the measles virus group in Zurich did try some positive strand mini genomes, and we tried with the full length, so it worked very nicely.
So what made you think of trying the positive strand? Was it something that you just said, let's try it, maybe it'll work, or was there some logic?
Yeah, I don't really remember, but looking at all those great groups, there were big groups, very, very famous researchers who failed. So that told you something must be wrong, something simple, and just turn around.
Had you expressed the minus strand for influenza?
We've done it any which way.
Any which way it worked.
It works with both ways, yeah.
So why is it different? Why don't we get the same issue? Do we know?
We have smaller segments, and then you have a helper. I mean, this was only true for the helper virus. In the system where there was a helper virus present.
And certainly the nature of the RNPs is also a very important point. In the recent years, the ribonucleoprotein complexes, the structures of these complexes came out for VSV and also for some paramyxoviruses now. And the RNPs of these mononegavirales and rhabdoviruses in general are very tightly organized, and the nucleoprotein makes something like a clip where the RNA is not accessible. And that is quite different for influenza virus, where the RNA is sometimes outside of this complex. So this is more highly structured, more structured than probably the influenza virus RNP. So you were lucky.
Luck is always good. We were lucky too.
For your rescue, you just need one plasmid?
3 plasmids
So you also have the helper ones.
So 3 plasmids is sufficient? Now, I thought I saw something where...
Usually the standard procedure is 4 plasmids, where you have the viral antigenome RNA, which encodes the viral antigenome RNA, and you have the nucleoprotein, phosphoprotein, and large polymerase protein, and that together makes you the RNP associated with a necessary polymerase complex.
So what did you mean when you told me you were making a plus strand virus out of rabies. Is
Okay, so that's not really the truth, of course, because these viruses will stay what they are. And you know, those positive strand RNA and negative strand RNA were initially distinguished by the ability to make infectious virus after transfection of their RNA into a cell, was described by Baltimore, and for positive strand, and the RNA from negative strand viruses was not infectious. So what we have now done is to make an RNA of rabies virus that can express all the helper virus proteins and simultaneously serve as a template for encapsulation into an RMP. So this fulfills, in these terms, the criterion of a positive strand RNA virus. So if you take the RNA, transfect it into the cells, then there should be virus created.
And this is a plus-stranded molecule that you put in the cell.
Yes, this is the antigenome of rabies virus.
Interesting. Interesting. And this virus is replicating fine. There are no defects.
Yeah, it has defects, of course. I mean, all those viruses are heavily optimated and adapted for good replication, which means that how they are, that's the best way to do it. If you change, and we have inserted iris elements from picorna viruses into the genomes, and that is stuff that they usually cannot need for replication or don't need. They are bigger and the translation levels are different, so there is an attenuation if you compare that to wild-type rabies virus.
Yeah,
this is optimized.
So these are not likely to arise naturally via recombination between different viruses because they're not fit.
No.
They're not fit
This,
enough.
this is an artificial system, and potential use of these is if you want to, for example, reduce the level of transfection plasmids that you need. This is reduced to 1, and you probably could imagine in introducing such one sequence more easily into a mouse, for example, and then can turn on, turn off.
It's conceptually very interesting.
I think so, and as you have heard in the talk before, many neurobiologists are interested in these rabies virus experiments, because rabies virus exclusively travels via synapses. You can map the connections of neurons, and there are a lot of great neuromice where neurobiologists have created to turn on and off genes, and the combination of these mice with a genetically encoded rabies virus tracer would probably be very, very promising.
Probably heard the name Richard Axel. He's a Nobel laureate. He won the Nobel Prize for discovering scent receptors, and he's a colleague of mine. So one day he had parked his car at the same time I did, and he was coming out and he said, "Vincent, I have to talk to you about rabies virus one day, because they want to use it to stereotactically put genes, I suppose, into specific areas of the CNS." I said, "Well, I don't know anything. You should talk to Klaus about it." But yeah, you're right. It's being used more and more to do tracing, which brings me to a question, and maybe our listeners are interested. Is it dangerous to work with rabies virus in the laboratory?
Not in the laboratory, especially if you do student experiments with rabies virus. We make sure that everybody is vaccinated against rabies virus, and that is a case where vaccination works very, very nicely and effectively. And we are these days only working with viruses where there are good vaccines available, rabies and measles virus, so you're on the safe side.
And so vaccination protects, and there's never been a laboratory-associated accident?
Never.
That's good, because
Not from a vaccinated person.
Are your strains wild type? They would be lethal otherwise?
Wild type, we are mostly using a vaccine strain,
right?
You know, and this is an attenuated live virus, which has been used for vaccination of wildlife here in Europe. And this is an example where not only patients are being vaccinated, but also the transmitting animals. And this virus was used for eradication of rabies virus in Germany. Germany was declared rabies virus free, I think, last year, and they distributed those live viruses in baits from the helicopter, and that was a very successful trial. And the virus is attenuated, and there is no rabies case observed by this virus.
We do a similar thing in the U.S. We drop rabies vaccine in various kinds of baits or packages, and the animals pick it up.
This is interesting, not a virology story, but rather an ecology, physiology story. I started in Tübingen working on rabies virus because they had some money earned with this vaccine, and they wanted to do some molecular biology, and their problem was then to make the right baits. And apparently foxes take fish meal baits very nicely, they chew it, and the biggest problem in rabies virus, however, is dogs. They are unlike foxes, they do not chew on the baits, but they swallow everything. And same for gorillas and monkeys. If you think of Ebola vaccination against gorillas, they like some baits, special baits, which taste for mango, they like best. So you get into various problems when you thinking about delivery on rabies virus vaccines.
In the U.S., there was a story not too long ago. I believe they vector some of the vaccines in a vaccinia virus backbone, not vaccinia, but some pox virus. And a woman had been out picking berries, and she scratched her hands on the berry bushes, and then her dog came back with one of these baits in its mouth, and it had broken, and she took it out of the dog's mouth, and the virus got on her cuts, and she ended up with a vaccinia rash because of it. And she was, on top of it, she was a bit immunocompromised. But it is interesting ecology story. You know, you put these baits out there, and things can happen.
Sure.
Anybody have any questions for us? I have a nice little magnet you can have here. I have 4 of them to get rid of. Yeah, Stefan.
I have a question for Professor Parisi. So, I mean, for us, of course, it would be nice to get just one shot of influenza vaccine, but I think for the pharmaceutical companies, it's probably more beneficial to produce every 2 years new vaccine. So are you going to open your own company, or what's going to be?
I think clearly if you make a better mousetrap, if one makes a better vaccine, then it will be widely accepted. So maybe worldwide 5% of the population only have access to influenza virus vaccine right now. So if one would really achieve a better vaccine, one which lasts longer, I think there will be a great push to accept that from all the companies.
Are you so optimistic, or would you think, I would say that a more realistic process is that somebody, a charity like the Gates Foundation, gets in and finances? Are you thinking that the companies will really work against themselves and put themselves up?
I think if it really works, I think the Novartis, I mean, I don't think they're that short-sighted. I mean, as I said, there would be a much bigger market, 20 times as much. So that would be, if they could really make a vaccine which is available for a lot of people, and I don't think there would be any hesitation by industry to accept it. I think that is, I don't think as badly, or so to speak, in terms of the companies. I think if there is a better product, they will use it.
Do you think that by developing a new kind of vaccine against the stem of AJ, will not this introduce new types of stems against which you have to develop new antibodies again?
Can you repeat the question, Peter?
So if I repeat the question, so if we make a vaccine just consisting of the stem and only make and induce an immune response against the stem, wouldn't the virus be smarter yet again and change its stem? And that is possible. However, the stem, it may be harder, more difficult for the virus because of the structure which is associated with that. And also fusion is involved in all of that. So it may be a little bit harder for the virus to evade that kind of immune protection. And it hasn't changed over the last 60, 80 years where we have viruses and have all the sequences. That region has been very much conserved. So that is probably, the virus is probably not at liberty to really change it as frequently and as effectively as it is the more flexible upper part of the molecule.
And what do you think? How many shots will we need of this vaccination?
Look, ideally it would be one shot, yeah, one administration.
But do you think that's realistic?
I think it is possible. Yeah, I mean, I think at this point, I think one would need an adjuvant, but there are, as Dr. Raccaniello mentioned, there are new adjuvants available, and I think it's realistic to hope for a vaccine which is adjuvanted and will be.
There you go.
Thank you very much.
You get one for being up here too.
Then I have two.
Two. Why you get two? One for being here, one for asking a question. All right. I have many more at home. I can get you more. I'd like to read a couple of emails now to show you the breadth of the kind of listeners we have. The first one is from Simon, who writes, "Thank you for your podcasts. I subscribe to both TWiV and TWiP, so we also do a monthly podcast on parasites called This Week in Parasitism. Find them fascinating. I work as a truck driver here in England and listen to your podcasts whilst driving about. Currently, I am studying for a life sciences BSc with the Open University and just coming to the end of my first year. I've just finished a module on microbes, and your podcasts add to the stuff that I've learned doing that. During the TWiV and TWiP, I find the questions Dick and Vince ask each other very helpful for explaining things in a more layman-like way. Hopefully, as my studies continue, I'll be able to keep up with the more complicated aspects of the subjects." And then he also indicates that I post my lectures from Columbia on iTunes University, so he's also downloaded those and listened to them. So this is amazing, right? What is Open University? Do you know?
Basically, you do it from home.
Home, okay. Well, that's great, Simon. Thanks for writing. The next one is from Josh. I emailed back in early 2009 when I first found your show to tell you how much I had been enjoying it. Since then, I've listened to all of your episodes. I'm a medical student at the University of Dundee in Scotland. In the UK, the medical course is undergraduate and takes 5 years. After listening to your show and hearing about the interesting lab techniques from brilliant researchers, that must not be us, of course, I decided to take up the university's offer of doing a BMSc in a single year before returning to the medical course. It is very challenging to go straight into the final year of a course that medical students are taught little about, but I decided it would be worth it for the practical lab experience. I've just begun my pharmacology degree course and have a placement in a lab working on diabetes. I've already treated cell cultures with various drugs and have just completed my first Western blot. Having never done any lab work before, I've heard you guys talk about including PCR. Wait a minute, I skipped a line. Having never done any lab work before, this is all very new to me, but I'm really enjoying it. I'm looking forward to using various other techniques I've heard you guys talk about, including PCR. Just thought I'd let you know that you are the people who inspired me to do some pure science. Hopefully someday I'll get to work with some viruses too. So in my view, there's nothing better we can do than to inspire people.
My BMed Sci, because we run the same course, so we're not far from Dundee.
Really?
My BMed Sci students work on viruses.
Maybe. Josh.
The thing is, though, they get a degree in 1 year because it's intercalated in their undergraduate medical course. So they get a degree in 1 year. So
wow.
The scientists kind of don't like that.
All right.
But they're super clever. So.
That's amazing. 1 year, yeah. They had these two, the truck driver and Josh, both had HONS after this degree. Does that mean?
Yeah, honors.
Honors?
Yeah.
What does that mean?
It's like you got to do an honors option. Usually it's an extra year.
I see. All right, this one I think is for you, Peter. It's from Eric, who writes, since influenza virus has 8 genome segments, and each segment carries a gene or genes that are vital for the virus to replicate, how does a budding virion guarantee, and then he says in quotes, I know how you feel about anthropomorphizing viruses, but I can't think of a better word, that it will contain all 8 genome segments. Is it simply the ones that are missing 1 or 2, or have doubles of a segment will not survive, while those that by chance just happen to have all 8 will proliferate, a sort of survival of the most complete genomes? Or is there a different, more sophisticated method that the virus employs in order to get all 8 genome segments into all or at least most of the budding virions?
So it's an old question whether the virus is really smart enough to package all 8, 1 through 8, and only those 8 to make an infectious particle, or whether it's random that it can package more than 8. And it looks like that the virus really is smarter than we thought, that it knows exactly which virus, which RNA to package, and that it gets the correct 8 RNAs, a full contingent, into its envelope, and that there is a very sophisticated packaging mechanism which we unfortunately have not been able to elucidate completely.
You know, I remember when I was in your lab in the 1970s.
Don't say it.
It's okay. So this was a big question back then, and Bernie Fields visited you once. And he came, Bernie Fields, of course, of Fields Virology, he came striding into the lab and he said to you, I know the experiment to figure out how reovirus puts 10 segments into the particle, but he wouldn't tell you what the experiment was. That's stuck in my mind ever since then. But he never figured it out back then either. So the next one is from Jamie, who writes, knowing how much you guys love bats, and on the off chance you haven't seen it yet, I thought I'd bring the following to your attention. It seems deforestation in the Amazon is causing large-scale vampire bat attacks on the local population of Peru, which in turn has caused an outbreak of rabies. How's that for medical ecology? So she gives a link to an article at BBC about this. It's only a short news article, and I'd like to see more information, but it seems to me that if these bat swarms are spreading rabies, it's quite probably they're spreading other nasties as well. Coronaviruses spring to mind. It doesn't mention that they are checking for anything else, and obviously vaccinating the bitten for rabies is a priority, but it seems like an ideal opportunity to measure bat-borne viral spread to humans. And she says in a postscript, originally I wrote, knowing how batty you guys are about bats, but I had to change it. I made myself groan. So, Klaus, do you know anything about this?
Sure. I mean, bats are reservoirs of many viruses, including coronaviruses, those novel viruses, Nipah, Hendra, and also rabies virus, of course. And that is known for some time. There are, here in Germany, rabies-related viruses of the Lissa virus genus, which are somehow adapted to bats. It appears to be they are adapted to bats, and they are circulating in the bat population for tens of years or longer. And if I remember right, there was one case reported that was caused by bat rabies virus, and the more strange thing is that you in the U.S., you are you're such kinds like silver hair bats, so not vampire bats, but insect eating bats can be infected by rabies virus, and in fact rabies virus cases from silver hair bats were not so rare in the U.S. So bats are a common source of virus infection, could be a common source of virus infection. Rabies itself in Europe is probably not a problem, but in the US, when certain species transmit those classical rabies virus, when bats adapt these classical rabies virus, that may be dangerous.
So it's interesting that when you disturb the bat populations by deforesting, then they impinge on humans, which is a very typical pattern of what we do. We mess things up and then we pay for it one way or another. The next one's from Bradford. This is actually a question for everyone. He says, I just want to tell you how much I enjoy listening to TWiV. I couldn't imagine my commute without them. So the next time you see someone on the subway with earphones, earbuds, they're probably listening to TWiV. I currently work in the virology department at the American Type Culture Collection in Northern Virginia. I spend my days in the lab replenishing stocks of just about every kind of virus and enjoy every minute of it. My question for you is, when you're doing your research, where do you get your viruses? ATCC, being a bioresource, provides many different kinds of authenticated organisms to researchers and universities around the world, but the vibe I got from my lab leader was that most resources in the field of virology tend to come from neighboring labs or researchers doing similar work. I'd be interested to hear how you guys get your biologicals in the academic community and how you make sure what you get is authentic. Thanks, and I look forward to many more TWiVs to come. Well, speaking for myself, I certainly get viruses from colleagues quite often. But now and then, if someone doesn't have it, I will get it from ATCC. What do you do, Peter?
Same thing.
Yeah. You get a lot of clinical isolates, I would think, also for flu.
From the CDC.
That comes from
yes.
CDC.
Yeah.
So when you get a virus from a colleague, you have to depend that they're giving you the right thing. How many times have you got a plasmid from someone and it's wrong, right? It happens. How about you, Andrew? Where do you get your viruses?
If we get it from somebody else, I always make sure it's sequenced.
Ah.
Unless it's too big, you know. But plasmids, get them sequenced because you know
Yeah,
your
plasmids,
best friend might have sent you something that's not
Well,
right.
with sequencing being cheaper now, you could sequence even adenovirus, no
Yeah.
problem. Right, so that's not a bad idea. But you may have, yes. Come. Why don't you come here and use the mic?
So that's something positive for ATCC, because when we started to work on cytomegalovirus, mainly mouse cytomegalovirus, we ordered the virus from ATCC and we got it. And later on, since we have been discussing here the principles of plasmid cloning of viruses, and we later on achieved the trick to clone herpes viruses as plasmids. And many years later, by having cloned several herpes viruses, we found out that our Smith strain, mouse cytomegalovirus, where we started from, somehow had a defect. It didn't really have the property we were expecting. And so we didn't know whether in our first cloning we made a mistake and we lost something, or whether it was a property of the ATCC virus tube. And we ordered it again. And then we are finding out that, which is also quite common among viruses, even if they are cloned by normal procedures, they are not really cloned. You always have several sort of individual DNA species, and they're heterogeneous. So we went back to ATCC, found out that it was a polyclonal population. By making a clone, we got one of those, which was only sort of one representative, but not representative for the complete clone, and we found that's typical in many of these cases, and it's nice to have such an institution where you can go back to it and study and see what you originally have been using, because in the lab these records are often sort of not kept as they should be, as it is in ATCC. So thank you.
Thank you very much. Good story.
I mean,
But you.
a very nice source if you are working on RNA viruses is just in case of those that are available as vaccines, it's just to go to the Marienapotheke, the Sendlinger Torplatz, and to buy, for example, a measles vaccine or a mumps vaccine. So that's what we also did already. And these vaccines are very nicely characterized, very nicely produced.
Yeah, so there you go, Richard. No, not Richard. That was Brad from ATCC. By the way, ATCC, as far as I know, is supported by grants and donations. It's a nonprofit organization. And I've heard people be critical of them. They're very expensive because it's a nonprofit. For example, I was just looking at a cell line. It's $450. But then again, it's the right thing, and you can freeze down aliquots, obviously. Sure.
But especially for the steroids, I think institutions like ATCC or in Germany we have similar institutions are absolutely necessary, because cell lines, if you get it from colleagues, are very often not what you want, what they pretend.
Yeah.
So I mean, this famous HeLa was going around, the HeLa cell lines, and it was taken as many different cell lines, and everything was HeLa.
Sure.
And the same is true with the Bergen conformal lines also. So these institutions like ATCC, they spend a lot of effort to characterize in detail and to genotype. And this is something which has to be done, which has to be requested by journals in general.
So there's an analogous organization in Germany.
Yes.
What is it called?
Deutsche Stammsammlung für Mikroben und Zellkulturen oder irgend sowas.
Glad you asked.
It's DSMZ.
DSMZ.
Z.
Okay, DSMZ. I have to admit, I like the German too. I like having it on TWiV. Okay, let's do a couple more, and we have to give them some time to rearrange this room here for the next talk. The next one is actually from Richard. He works in a sewage treatment plant, and he has isolated phages from the sewage. He's sort of like a, an amateur virologist as a consequence. He's grown up phages, he's made plaques, and he said he can get unlimited amounts of phage from the sewage treatment plant, as you would know, because our intestines are full of viruses in the bacteria that are resident there. So he has a few questions. If phage can be selected for and very quickly wipe out a particular bacterium, why are they not used more in medicine? It seems that it would be possible to treat bacterial infections this way and reduce the use of antibiotics. This would be more sustainable since the bacterium and phage are locked in an evolutionary relationship. Resistance should be much less of a problem. I'm sure there must be a reason this isn't done, because it seems almost too easy.
This is being done, and if I remember right, since a long time in former Russia, but now I think also here in Germany there are companies that are using phages, at least for disinfection of surfaces, and probably also for further kills, if I'm right.
And I think the problem in people is that they're antigenic and you clear them rapidly. So therapeutically they may not be useful. That's my view.
Good if you want to decontaminate
Yeah.
the bacteria, as Klaus mentioned.
Sure.
Food treatment is perfect. I know the companies actually taking the subject up.
Right. But in people, though, is that the correct answer, that they're just cleared too quickly? Now, the next question you're all going to love. With the recent news on synthetic life, do you see a future for synthetic virology? And if so, in what fields? I would imagine gene therapy would be one possibility, but are there others? Of course, we all do synthetic virology.
That's the point. We are doing synthetic
Yeah.
virology.
Yeah, it's been, in fact, it was, if you consider viruses organisms, then the first organisms synthesized in the lab were viruses. And it was done with a lot less fanfare than the Craig Venter experiment. In fact, I think David Baltimore pointed that out in an interview that he did. So yes, Richard, we've been doing it for the longest time. It was first done with bacteriophage in 1978, Charles Weissman, and then poliovirus in 1981. The next one's from Levi, who is a pre-doc at University of Florida. And he sends us a link to a BBC News article, which is based on a Nature paper, about a vaccine patch that delivers a vaccine through hundreds of tiny dissolving polymer microneedles. They barely puncture the skin and are supposedly painless. Yet a study in Nature Medicine claims that they induce a better response against influenza than a vaccine delivered by a jab. Do you think this technology could ever see common use, and could we ever use it to vaccinate ourselves rather than going to a doctor's office or other vaccine clinic? I know many people who don't like needles who would love something like this. However, there are also plenty of people who I'm sure would love to sue a manufacturer if they used the patch but still got sick. Would self-administration be a realistic goal for future vaccines, or will we still need someone to apply the patch on our arms to make sure we did it right? Thanks. As always, TWiV is excellent. Keep up the good work. Know about this? What do you think?
Yes, it's a very interesting technology. Basically, it is, as the letter writer said, it is consisting of a number, so it's a small patch which has a number of these spikes in there, and they are pressed on the skin, and they are going deep enough to actually reach dendritic cells. So that's sort of what's happening, is that there's a contact made, and the vaccine is either incorporated into these needles or is dipped into it. So once one puts that onto the skin, one gets these small pricks, and there's enough contact with dendritic cells. And that is, I think, is a very reasonable and very, I think, promising technology. Yeah? Interestingly, the material is a little bit in question still, whether these are steel pricks or whether one can even have plastic, basically, which then dissolves as it is being put on. So there is a lot happening in that respect, and I think it's a reasonable approach.
But is the virus stable on this patch?
There's certainly a half-life or a shelf life of a limited time, but it may be 6 months or a year.
Okay,
that might be
quite
fine.
long.
Yeah.
So you could imagine this as a prescription you take home and you apply it yourself?
Correct,
That would
yes.
be reasonable.
It's like a scopolamine patch
Yeah.
for against car sickness or air travel, etc.
All right, let's do 2 more quick ones. One is from Katie, who is a second year. She says she's a lowly second year PhD student in Montreal, Canada, but no PhD student is lowly, of course. I just wanted to tell you that you are my heroes, and I wish, as do many of my student counterparts, I'm sure, I could hang out with you guys during TWiV, not to contribute, obviously, merely to absorb it all in person. So I couldn't resist reading that. I like being somebody's hero. That's great. We do. And one more. Let's see. This one is from Eric. So Eric is Eric Donaldson. He's a assistant professor at the University of North Carolina. He's been on TWiV twice. And he actually just published a study in Journal of Virology. They trap bats in Western Maryland and sequence the virome in the guano, in the bat guano, and they find basically every virus that you would want that is in bats, especially a lot of insect viruses, of course, because they eat a lot of insects, but you find rabies, coronas, even herpes viruses. So that's in JV accept. But Eric writes, after the last time I was on TWiV, I received an email from a high school biology teacher in Hong Kong who was interested in helping with the viral metagenomics project. So I sent her a data set to work with, and she's blazing away. In the meantime, we've been discussing how we can bring her high school students into the project, and she had some really great ideas that I hope we can implement in the next couple of months. So hopefully we will have students at all levels hunting for viruses in a haystack of sequences. Once we have something established, I intend to reach out to our local high schools too. I think this story illustrates how widespread TWiV's influence is and demonstrates what a powerful tool it is for science education. Thanks for your continued effort in making this podcast a success. So one of the things Eric said is that the data takes weeks and weeks to sort through, and if people he can outsource it by giving it to people and using their computers to look at it. And this person in Hong Kong found him because of us. So another example of the power of this new medium. And I hope that by seeing us do this today, that someday in the future when I'm no longer here, that some of you will take on such efforts yourself. Now, the last thing we usually do on TWiV is what we call a science pick of the week. We like to pick things that are science related that we want to share. It could be a book, a movie, or an article. And I've asked Katerina to do a pick with me. And so while you're getting yours ready, I will give...
mine.
You have yours ready? Would you like to tell us what you picked?
Shall I start?
Sure.
Since we're in Munich, I thought I'd take something Munich related, and I've chosen, so my colleagues in the SFB, they know it because they're also in Munich. I've chosen a museum. It's called German Museum, and I think it's Germany's most popular museum and the world's biggest museum on natural sciences and technology. And what I like most is that, so it's like a big playground. You can touch everything. You can do chemical reactions. You even can clone your DNA. And so you can use a pipette and everything. So, and I have the feeling that when people go there, then they do understand better what we are doing every day. Because then it's not pipetting liquids from A to B, and at the end you have something magic, but because they see what they are doing. And I think I checked the homepage, and most recently they opened a kind of DNA laboratory, and there you can do genetic fingerprinting, cloning, ELISA, everything. It's quite cool, and what I like is it's assisted by young scientists. So sometimes the PhD students do it to get some more salary. And so you learn how to teach, and then you have I think it's quite nice and quite informative.
Sounds great. It's a perfect kind of pick. So our people in Europe will love it. And I'll get that from you so I can put the link in our show notes. So my pick was selected by Judy, who is a high school science teacher. And she wrote, "Hello, gentlemen, and soon more ladies of TWiV. Every December, the Howard Hughes Medical Institute does the holiday lectures on various topics in science. The previous ones have been on evolution, molecular clocks, and the metabolism of fat. The ones this year given by webcast are on dengue fever and emerging disease. You can watch them online, or you can get a DVD, and educators can get the DVDs free of charge. This should be a pick of the week, and I agree. That's a great pick. This year's lectures, Viral Outbreak, the Science of Emerging Disease. Watch two leading virus researchers explain how they use both simple and sophisticated technologies to detect and fight infectious agents. Join Joe DeRisi and Eva Harris as they discuss their strategies for combating today's epidemics and preparing for those of tomorrow." So that is my pick. The Howard Hughes Medical Institute will put a link for that in the show notes. And that will do it, unless anyone else wants to recommend something. I didn't ask you guys to do it, but anything, any of you read a good science book lately? No, huh?
I read the Healer book. You know,
you read
there's a
The
recent one
Immortal
came out.
Life of, did you
Henry
read the acknowledgements in the back?
I did. I can't remember. Oh, yeah.
I'm sorry. I'll cut that.
I hadn't met you at that point.
I'll edit that out. The Immortal Life of Rebecca Skloot. Actually, I was just at the University of Pennsylvania. The Division of Infectious Diseases has a book club every month. This is very interesting. All the ID docs sit around. They pick a book and they discuss it. So they picked The Immortal Life. What a discussion. It was just amazing. And in fact, I recorded it. It's going to be a future TWiV episode. But they had a variety of people who are involved in medical ethics and people who study the cultural limits of medical practice, how it applies to different cultures differently. And it was a very, very animated discussion. So look for that on an upcoming TWiV. You can find TWiV on iTunes at the Zune Marketplace, or you can listen to it on your smartphone by Stitcher Radio. It streams it live to your smartphone. That's a free app for iPhone, iPod Touch, iPad, BlackBerry Palm, or Google Android. Or you can go to twiv.tv, where we put the episodes. You can play them there or download them, and we keep show notes there as well. And if you like TWiV, well, you guys who haven't listened, you should check it out, because I think you'll like it. And tell your friends. If you use iTunes, you can leave a review there. We're usually on the front page of the medicine podcast section, which is really great. And we stay there because we have a lot of comments written. So if you haven't done so, go over to iTunes and just leave a sentence or so, and that would help us a lot. As usual, send us your questions and comments to twiv@twiv.tv. And I want to thank everyone for being here today, starting with Katharina. Thanks so much. You feeling better now?
Yeah.
All right. So the next one will be easier, right?
Yeah.
Yeah, you see that?
We can do it again.
Great. Katarina is a postdoc in the lab of Dr. Ann Krug at the Technical University of Munich. Good luck in the future.
Thank you very much.
All right. Peter Palese, professor and chair, microbiology, Mount Sinai. Thanks for joining us.
Thank you.
Thanks for making me a virologist.
Good.
And Karl Klaus Konzelmann, professor at the Ludwig Maximilian University right here in Munich. Great having you.
Great to be here.
And good seeing you again after many years.
Sure.
Right. And Andrew Baker is Professor of Molecular Medicine, University of Glasgow. Nice meeting you.
Pleasure. Nice to
And
meet
thanks
you too.
for telling us about that work, and we'll hear it again in
Hopefully.
about a half an hour, right? And of course,
Yes.
special thanks to the junior faculty of the SFB 455. Can you say that again, SFB in
It's
German?
Sonderforschungsbereich.
Ah, wonderful. Thanks to the SFB 455 for having TWiV live in Munich. And I want to especially thank Stefan Jordan and Jens Losing for inviting me. You guys wrote me the email originally, and particularly to Jens, who took care of me, made sure I got everything, and really helping and finding a cable at the last minute. So thank you very much. And see you in Princeton, where you're going to do a postdoc. I also want to thank all the junior faculty of the SFB 455 for organizing this fantastic meeting. You're the future of virology, and thanks to the audience. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.